Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Viewers, welcome again to this session for the series related to home learning via TV. So in today's lesson, we are wrapping it up, but this time in a different way, with the questions and what is expected of you as a candidate, as a student in class, to pay much attention to every time you come across questions. So we look at these as command words. We call them command words because they're instructional. They tell you exactly what to do when given an exercise, when given an assignment. So this is going to be a unit assignment. Remember, we're doing Unit 5 about cell and nuclear division, and this is meant for S5, but it does not mean that the S6s just relax back. You also can benefit from this. So these command words do tell you exactly what is expected of you every time you sit in an exam. Where is your focus in terms of attempting the question? How far do you go, and where do you focus? So we have common words. We meet in questions like describing, discussing, giving an explanation, a reasoned explanation as to why. We also may suggest. Some of the questions might ask you to suggest, as in suggesting as how many answers can look different, yet all correct. And are we able to evaluate? Are we able to make a judgment? Yes, we have this that we are looking for, but alongside, you have the negative part of it. Are we able to weigh the benefits against the disadvantages? Are we able to make a comparison? When we do compare things, are we looking for similarities or we are looking for differences? And this is where learners have always made errors. In comparing, we are looking for what is cutting across all of these things we are trying to relate with. So they basically can be looked at as similarities. And then a contrast, definitely. A difference, a very sharp one. So we shall see some other questions coming up with different command words being embedded in. The number goes on. It's non-ending. But those are a few I've been able to capture in and focus so much on in the unit. So, like we have already said, when questions are surfacing on this series, it is always important that you have your camera close by, phone, anything you can use to capture. For those who have decoders that are able to record, then press the record button, and then you're able to have a review of your lesson. So let's look at what we have. Remember, your camera should be ready and be in position to take the first shot on question number 1. What do you see? What goes into your head right away from the time you look at the question? It is that capture, it is that first impression you create when you look at the image on the whole question that is going to tell you, yeah, I think this is coming from photosynthesis. Then if you don't see anything green or chloroplast, then you just, I think I have to focus on this chapter. Then that way, your small pockets in the head, in which every component is stored, some are able to lock, and then you unlock one where it is stored. Are we able to identify? Can we give a reason for identifying? They're saying, 1, whether the cell is a prokaryote or not. Remember, the stake, the score, against the question will always tell you what you need to think about as you answer. 2, they're saying part of the root tip or a fingertip. This or a root tip. Now the question should be, how do I categorize these 2 forms of tissues, in the fingers and in the root tip? Is my finger a meristem? Is it a growing end? Is the root tip they are referring to a meristem? And if it's a meristem, what is the level of activity in that? what should be evident that when I'm given a photomicrograph of a root, it should tell me exactly that, well, according to the stages of division, then I think I'm at this stage. If we say meristems are regions of active mitotic division, is the cell really telling me that there is so much mitosis, or it is sleeping, like we said, resting? The magnification of the drawing is 2,500. The times here is a sign that we are enlarging. So now the mathematical component comes in. Do you recall the formula? How do I get exactly what they're asking for? How do I calculate how long a 5 micrometer scale bar should be if it was to be added to the drawing? Now they are looking for your skill in terms of drawing, magnification, calculations. I mean, those scientific skills that are required in microscopy. And then predict. So we have 3 interesting words in here. Are we able to identify? So we should tell. There must be naming here. 2, are we able to give a reason? Can we tell why? Can we give an explanation for our choice? And then the other part here is mathematical. Numbers, calculating, skills. And the third part, prediction. Are you able to foretell? Sinavuze ngo umuraguzi cyangwa umufumu abandi ibindi muhita gutera. Foretellers. You are not practicing African chemistry here. So you base on what you have as a concept about a topic, and then you're able to predict what would happen to the cell if it was placed in a concentrated, wonderful part. In assessment, don't zero your head and your mind onto one topic. These things cut across. They say, they are going to ask us about cell division, as if they are never going to ask you to label a cell part. Think about it. The other one is about data. I love this. I hope you've taken the caption, and you should be able to look at these graphs. But what do these graphs tell you? You have the number of cells in thousands. So you look at one here, then it is 1,000. 2, then it is 2,000. 3, then it is 3,000. And so on this way. DNA. Per nucleus of the cell. And they are giving us units in terms of amount and the number of cells, and also the parts. So you have 1, 2, and 3. Rapidly dividing cell culture and non-dividing cell cultures. Now you need to think, why these different bars? Why is the question set like that? So, they're saying that in sample B, you need to be able to make a deduction. You deduce which phase of the cell cycle. Now you have to start thinking, hi, cell cycle. We just finished meiosis and mitosis. Do I even still remember interphase? What about the G, S, and then G1? But the good thing is that they have brought these numbers back to you, so you can click from there and start off. So the deduction here is going to be telling us in terms of when we look at the amounts of DNA. When do we have the amount duplicating? When do we have it doubling? When do we have a lot? And when do we actually have numbers at a certain amount? Is it when the cell is dividing? Is it when it is in interphase? Is it when it has duplicated? Think about it. So that we're able to tell that, oh, G1, oh, G2, then S, what exactly happens in S? that I'm able to look at the amounts and check, oh, the same, and then here, and then this. What exactly is happening here? If this is a non-dividing cell culture, these cells are not dividing. So exactly what's happening? What could be the amount of the DNA if the cell is not dividing? What about when it's just gone out of resting, as we use the words, and then we now go to high level of metabolism, a lot of proteins made, then S phase, so much DNA replication. Do we have doubling the amount from 46 to 92 before we again go back to 46, 23, 23? Something like that. And then B, they're saying that we need to make an estimate, the approximate amount of DNA per nucleus that would be expected in the following human cells. Wow, I love this. Do we know what exactly the amount of the DNA is in prophase and telophase? What about during interphase? When interphase is over and S is complete. Now the question should be, do we need another duplication? Do we need another S? All right, that's yours. So the figure is there. Look at the height of the graphs, this and that. Why do they look the same? And then we now have 2 and then 3. What exactly has happened? So if we were to go to this by telling us to make an estimate, they are saying, wonderful part of it, human cells. Can this be a hint for us to think, if they say human, what's the chromosome number? So when we talk of somatic cells and germ layer of cells for gamete formation or reproductive cells, do I need to think about the chromosome number? Yeah, something like that. Which takes us to the next. Figures. Figures keep coming. Because they tell a lot and they carry a lot more information than just the text. I hope you've finished reading the question. Look at the background information to the question. Life cycle of humans and mosses. Whoa. Plants and animals. Showing both their haploid numbers of chromosomes and the 2N to represent the diploid condition, twice the amount. So the pairs. So we talk of 23 pairs, oh, that is 46 chromosomes in total. We talk about 6, then in total this would be 12. Spirophytes of mosses, now, remember your senior 4, remember your diversity, your classification, your senior 1 classification. And the main moss plant and consists of a stalk and capsule in which the spores are produced. Oh, spores. How do they form? And now they're asking us to be able to outline the similarities between the life cycle of a moss and of a human. Outline. So they are just short statements. An outline does not require you to write a lot. And look at the 5 marks given to the question. And then, what about distinguishing? Put differences. Can the picture here guide us and tell us what is happening here where we have the spore? And we don't have spores in humans. We do form male and female gametes as N. So the figure is giving us some bit of guidance on how to answer number 1 and number 2. I think we are good to go. Ish. Remember the command words. And something important we need to take note of as well, moss and human. mouse and human, plant and mouse. Are these same species, different species? What about in the form of the way they reproduce and stuff like that? So, the other one, what we finished in the previous lesson, about us, when we are still a fetus. That before we are even born, technology has given us a chance to be in position to tell, oh, yeah, they went for a penis search. I'm not saying that we have to go in trousers looking for them. No. It is a scientific description of how mothers can tell the sex of the child before they actually give birth. So they, in their shopping, are able to determine, should I buy clothes that are boyish or girlish? Is that language correct? Yeah, okay, baby boy and baby girl. Gender, I know you're there saying, gender sensitive. So we do have these cardiograms as diagrams as extracted from the previous lesson where we said that people are able to tell and then prepare. Now the question is, one, are we able to differentiate between these chromosomes? Are we able to differentiate the pairs? Check the numbers from number 1 all the way through to number 22 and then the 23rd pair. I love this. 23rd pair. Let's circle it and pay much focus on it. Could the designer of the exam could have intended that you actually focus on that? Is it part of the question? And this is where you as a candidate have to think about that, can I preempt the teacher's mind? If I have understood the concept, can I figure out in which way he's going to set the question? Then I'm able to right away look at the question. Before I even start getting the pen, the answer is already flowing in the head, and I'm already concluding before I actually begin. So, 1, they are looking at 1 chromosomes. These are pairs, could be homologous, but the question is, when we look at them, 1 is saying or asking us, which one is the longest, then the shortest. Distinguish between the structure of human chromosome number 2 and chromosome number 12. So can we look at the sets? Number 2 is here. And where is those? Here. Number 12. 12. Iyi ni ya mbere, iyi ni ya kabiri. Iyi ni ya 12. So, what are they asking us to do? Ese dushobora kuzitandukanya neza bitewe na structures tubona ukuntu ziteye. B, the human X and Y. They have even labeled them for us. Can we tell the difference between this and that and why? Before we even say, okay, I can see one is short, another one is longer. But are we able to tell maybe the position of the centromere in each of the two is a little bit different, or the height? Can we tell them in terms of the genes, the content they carry? How much do we know about X and Y? And then they're asking us finally, with a reason, can we tell the sex of this child from where this cardiogram was drawn? What do we need to base on? Which takes us here. Explain whether the karyotype shows any abnormalities. Wonderful. What we just finished the other day. That even when you've gone for some of these chorion sampling techniques and we need to actually check whether our children are going to be born with abnormalities or not, once we've got this map before delivery of the child, can we use these images before we see and how this child is going to be showing us all these observations as they grow? We see some kind of abnormalities on this child's body. Are we able to also tell from the picture? Which takes me back to this question. Every time you go for medical results, they give them to you in an envelope, you walk home and go, Mommy, here are the results. She took you to school to study. What's the relevance? That the doctor gives you results, you even don't ask the doctor what's going on, or you cannot even interpret some of the simplest things. That someone is given a cardiogram of his or her own content, maybe from the child or towards delivery, and you're still there as a mom expecting a child, or as a dad expecting a child, and you still say, doctor, can you tell us exactly what is happening here? Then I would have wasted my time talking about this. This is the relevance. So are you able to tell that the child is normal or abnormal? Check out and see. What do you think of this? Let's get it with a red pen and circle it. There. If we say the child shows an abnormality, can you give a reasoned explanation for that? Can you tell why? Can you support your answer? And then, what concerns most of us, here comes, in which we are looking at how old we have become, and then we are looking at the clock countdown, tick, 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 45. Some of us start fearing celebrating birthdays, so it comes as a surprise. And after I say, how old are you now? It's okay, I'm just old enough to know the difference between what is right and what is wrong. Hey, check this out. At some point, even when you keep hiding, you would have to start saying, I think I'm clocking towards 0. Not about to die, but you know what I mean. How many more years do I have before I can have a child? When is it going to be possible? Stuff like that. So look at the question very well. It is related to parental age and how non-disjunction can occur. When do we have these mistakes coming up? Can they be connected to age? What if I produce when I'm 50? What if I become pregnant when I'm 46? What are the chances? So here we do have a graph showing us the age, maternal age. Don't worry, the men, we shall deal with you next time. The incidence or percentages of all the live births. Trisomy, 3 chromosomes, that's chromosome 21, red line. All chromosomal abnormalities, green line. Now you need to check the occurrences. It's funny. Look at the hike in the graph and the age, but also before they actually kick 45, before mothers hit the period towards a session where they are unable to successfully give birth, where they are unable to successfully conceive. I'm not saying that others can push it due to modern technologies. Mothers at 50 have given birth, so worry not if you're still out there. We are behind you, don't worry. But the question is now, one, can you tell the relationship in the graph between maternal age and the incidence of chromosomal abnormalities in live births? Why are they even appearing at the age between 20 and 30? These abnormalities are coming up. Could it be that maybe we are even having more chances of ladies or girls getting married or producing before actually they even reach 30, that we have more of them, and so the chances are high. Increase the frequency, increasing the study, increase the sample space, then you have more chances. So using the data, calculate the probability that a mother of 40 years of age will give birth to a child with a chromosome abnormality. So we look at the numbers, start doing the math. If I am old to this age, maybe around here, what is the percentage that I'm going to produce a child with an abnormality? What is the percentage when I get married at the age of, say, 32? Do we always have this in mind every time we are to conceive and have babies? We just say, okay, even if it is at 50, no problem. But do you know that you are a clock? Count down. every month an egg goes, so by 45 you're virtually unable to successfully conceive. So if you push it beyond that time, and so we are now around here, what is the chance that you're going to have a child with an abnormality? What can we do to reduce these chances? Which takes us to the third part of the question as a continuation, that from the graph we are seeing only a small number of possible chromosome abnormalities are ever found among live births, and trisomy is much the commonest. Can we tell why? Remember the other picture we looked at in the previous lesson? Very few anomalies in childbirth, but among those few, the greatest frequency actually appears in trisomy, Down syndrome. Can we try to give a possible explanation and reason for that trend? How many are being born from mothers that are at a younger age? What about the old ones? So if you're trying to avoid the number in terms of age, though they say age is just a number, now the question is, why is it even when these mothers get married earlier at a younger age, still these trisomy cases continue to come up? And finally, can we have a discussion? Can we make our own argument for the risks parents may face when choosing to postpone having children? Family planning, but how far do you go? So in your discussion, you have to be like a double-edged sword. Cut here and cut here at the same time. What is the benefit? But what is the disadvantage? That's the discussion. You should be able to have an argument based on what you think is right, and in that right, what you think might come out as a danger. And lastly, take this snapshot quickly. The numbers are so many, as you can see, chromosome numbers, and read the question so very carefully. We are having species, plants, and animals, and you should be in position to look at the questions very, very, very carefully. Take your time. That's why I've made it the last slide, so that you are able to have your camera and take it, and then you can zoom in and be able to read the numbers and the questions very, very, very carefully. With that, as I wait for you to take it, off I go. See you another time.